Hello viewers, welcome to this particular lecture on malarial parasite and its control. Malarial parasite is represented by a species or genus Plasmodium belonging to protozoa. This particular parasite is actually developing in human, but it completes the life cycle in mosquito, that is female Anopheles mosquito. So in this particular lecture, I shall be considering the malarial parasite, the different types of malarial parasite, the life cycle of malarial parasite and its historical development, and also I shall be discussing the different damages that this parasite causes and the specific ways by which this disease can be controlled. So malarial parasite actually, mainly the common malaria is the Plasmodium vivax, which is the normal malaria or benign malaria. There may be also the Plasmodium malariae, which is also called the quartan malaria. There may be also the Plasmodium ovale, which is called the Tarsian malaria. And apart from these three, there may be the dangerous Plasmodium falciparum, which is also called the malignant malaria, and also the Plasmodium knowlesi, which is a zoonotic disease mainly invading the animals. And lastly, there is another type which is the Plasmodium hyoli, which is African malaria. Now all these particular figures you can see, this is the parasite or the merozoite of the parasite Plasmodium, and these are the RBC. So all these photographs are taken in a particular blood sample. So the best way to detect malarial parasite is testing of blood. And apparently this particular parasite can grow both outside the RBC, but mainly it will feed on the RBC, so it gets inside and it will produce different excretory matter inside the RBC by feeding on the hemoglobin. So basically it is a blood parasite which is surviving inside RBC for a long time, and also it can survive in liver. And apparently by seeing the different pattern of growth or stages of growth, we can actually identify it. So a very good histologist, just by seeing the infected blood slide, can tell you what is the nature of the pathogen, whether it is Plasmodium vivax or it is Plasmodium falciparum or any other species, right? This is a comparison of the different stages of malarial parasite, and that actually can be understood by the way they grow inside the RBC. Coming to the systematic position, as I told you, it is a protozoan pathogen, but nowadays it is also called Apicomplexa. It belongs to the class of Aconidiacida, and also it is having the order of Hemosporida. It is having the family of Plasmodidae, and the genus is Plasmodium, and the species may be Vivax or Falciparum or others. Now coming to the historical development of this particular disease. This disease actually was reported for the first time in 2700 BC in China. And not only that, people at that time could actually treat this type of fever with some hemp bark extract. So they knew the art of treating malaria with some hemp bark extract, which perhaps was some anti-malarial alkaloid. Much later, maybe that also, maybe in 500 or to 500 to 1200 BC, that was the time period, there was a severe infection all over the Roman Empire. It spread across Italy, Sardinia, and it was growing along the Tiber River. And so much dangerous was the fever, it was actually causing lot of death. And it was called, or it became famous in the ancient Rome as Roman fever, and still it is important in that name. Okay? Now, it was Charles Laveron in the year 1880. This person could for the first time discover that malarial parasite is surviving in human blood. And this particular discovery actually made him win the Nobel Prize in 1907, much later though. And it was Finlay who actually was thinking that this particular disease was having some association with mosquito. And he wrote a paper in 1881 stating that Anopheles mosquito is involved in it. But he couldn't prove it though. But it was this person who is Sir Ronald Ross, starting from the year 1894 till 1898, he worked in PG Hospital, Kolkata, and also in other parts of the country, and exclusively he proved that the female Anopheles mosquito requires human blood for the maturation of the egg, and by that way they transmit the malarial parasite to human beings. So in that way, human and mosquito, these are the two hosts of the malarial parasite, Plasmodium, where definitely he proved that the sexual cycle occurs inside the mosquito. So this particular disease or this particular pathogen is having a definitive host in the form of female Anopheles mosquito, and the alternate host where the asexual reproduction occurs takes place in human, and that made him win the Nobel Prize in the year 1902. Now that was in 1898 this discovery was made, and it was Walter Reed who in the year 1900 he confirmed the findings of Ross. And much later, this is a something different discovery in the period between 1917 to 1940. This person, that is Julius Wagner Jauregg, he showed that this particular parasitic infection can prevent a bacterial infection. So if a person suffering from syphilis was inoculated with malarial parasite, the disease syphilis could be controlled, because at that time, you understand, there was no suitable reply to syphilis infection. But definitely that was not continued though, because for treating one disease you cannot make a person suffer from another disease. Okay, this particular disease is growing worldwide, but you can clearly see it is mostly growing along the tropics. So it is a tropical problem. It is not a problem in the temperate climate. So it is growing all over Southern America, it is growing in Africa, and of course India. But the most important area where it is endemic and causing havoc is Africa. Number of death also is higher. And perhaps that is the reason also that Ronald Ross, in order to do research on Plasmodium, had to come to a tropical country, and that is India. So malarial parasite is invading multiple organs. Different organs are invaded. It can be symptoms like headache, it can have muscular fatigue, back pain, there may be chills and sweating, there may be dry cough, there may be enlargement of spleen, and there may be stomach nausea or vomiting. And at the same time, there may be sometimes some complication, and this complication may be acute or chronic. The acute one may be in the form of cerebral malaria invading the brain, or the chronic may be in the form of anemia, placental malaria, or severe jaundice. Okay?
Sir, why is jaundice associated with malaria?
You see, malaria, I told you, is growing on RBC. So the RBC is destroyed. So hemoglobin is destroyed, and hemoglobin will produce bilirubin. So the serum bilirubin concentration goes up, which is equivalent to jaundice, because the more number of RBC will be destroyed, there will be more bilirubin.
Okay.
So you understood? Yes. Good. Now coming to the life cycle of Plasmodium vivax. So as I told you, clearly you can see this life cycle is having the human cycle and the mosquito cycle. This human cycle, the mosquito is biting the human, containing these particular sporozoites which will enter the liver, and this cycle is called exoerythrocytic cycle or pre-erythrocytic cycle. What is the principle? This particular malarial parasite will undergo asexual division, producing the scisons, and these scisons will enter into the RBC and feed on the hemoglobin, produce a huge vacuole, we call it signet ring, and ultimately towards the end of this erythrocytic cycle, it will produce the gametocytes. And these gametocytes will be transmitted again to mosquito, where they will undergo sexual reproduction, and they will finally produce the oocyst, and again they will be undergoing reduction division to produce the sporozoites. So you can see that though the malarial parasite is spending more time in human, but yet mosquito, where the sexual reproduction occurs, is actually the definitive host. It is not the human, human is alternate host. Similarly, when we consider the life cycle of Plasmodium falciparum, you can also see that this infection is more or less the same. There will be the liver cycle, there will be the RBC cycle, there will be the gametocytes produced, and finally it will be in the mosquito where they are growing. But the question is, sometimes this particular parasite will cross the blood-brain barrier, will cause the infection, creating a fatal condition. So this Plasmodium falciparum or malignant malaria, it is so called because the way it is dividing, faster division, is also called cerebral malaria. This particular erythrocyte is being killed by that Plasmodium, and this is producing a huge particular vacuole. And this vacuole is not the part of the cell, but this vacuole is actually the part of the Plasmodium parasite, and this is called the signet ring. So an efficient histologist, just by seeing the pattern of the signet ring or the efficiency of the signet ring, can actually say which particular Plasmodium has attacked that person. As I told you that this particular infection is mainly in the blood. So what is happening? Ultimately, the malarial parasite is feeding on blood. So they are destroying the different components of globin and also the heme. So what do? Say for example, the globin fragments are converted to smaller peptides, and these peptides are ultimately utilized as amino acids. And these amino acids will be utilized by the mosquito, and at the same time there will be the heme protein. The heme protein will on one side produce the heme, and on the other side it will produce peptides. At the same time it produces the waste product. So this particular waste product, hemozoin, is deposited in the particular blood, and that hemozoin is toxic for us, so the body temperature goes up, and that results in chill and fever in humans.
Sir, I have heard there is recurrent fever in case of malaria. Why exactly is that?
You can understand this particular hemozoin production cannot occur once. It will occur, say, 3-4 cycles are there, and the moment the hemozoin is produced, then again the temperature will go up, the chill will be there, and it will be destroyed. And again the malarial cycle will continue. They will again invade fresh RBC, and this fresh set of RBC will again produce the hemozoin, and that will take some 1 or 2 days time. And that is the reason why this particular malarial parasite is classified on the basis of tertian, quatern or other varieties. Because the chill comes or fever comes, say, every 24 hours, every 48 hours, every 72 hours. This is because of this hemozoin cycle. Okay? So it is a cyclopropagating parasite. Clear to you? Good. Now, as I was telling, that in the life cycle, it is not really happy in the blood. It has to go somewhere else. It is adventurous. So this particular malarial parasite will get into the brain. It will cross this particular blood-brain barrier. What is blood-brain barrier? It is the barrier between the CSF and the blood. And you know this area is formed of astrocyte. So they will cross this particular blood-brain barrier and get into the brain. And the moment it gets into the brain, it needs huge concentration. So there is some strategy. What is that? They produce some specific protein EMP1 which will release the GPI anchored protein, and they will finally produce the adenosine. So the adenosine will help that particular parasite to adhere to the wall of the blood vessel. So that means, in fact, they will not be carried away. So the huge number of parasite concentration there is in the brain capillaries. And the moment they enter into the brain, they will cause serious infection, hypersensitivity, and that particular symptoms can be fatal. The person can get into coma, and that is cerebral malaria.
Sir, what is the mechanism of this malarial encephalopathy?
You see, encephalon is the other name of the brain. Because of the infection, there is some typical pathogenic symptoms developing, and this encephalopathy actually indicates swelling of this particular brain and the irregularity within the CSF, which also is accompanied by cytokine production, hypersensitivity, and ultimately resulting in death. So the basic mechanism is initiated by the crossing of the malarial parasite from blood to the brain. Now, apart from brain, I was telling you that this liver is a very important organ where the malarial parasite will reside. It is a repository for the malarial parasite, and this particular liver is showing enlargement, and the malarial trophozoites or sporozoites may remain in liver, may reproduce in liver, and they might develop conditions similar to jaundice or hepatitis or even sometimes liver cancer. So that way it is really dangerous that this particular liver which is a very delicate organ, which is the metabolic king of our body. All the metabolic activities takes place within liver, but this particular organ is getting affected because of malarial parasite, and our digestive power also is reduced. Now there are some people who are showing resistant to this malarial parasite. Who are they? People who are resistant may include the person suffering from sickle cell anemia. And this is how these blood vessels or blood cells will look like. That is, they are looking like a sickle. Instead of this round RBC, you are having sickle-shaped RBC. Why this occurs? Because you know this is a normal hemoglobin having 2 alpha, 2 beta chain. And in case of sickle cell anemia, you can see this is the deformity in the beta chain. Why this deformity occur? It is because of the 6th position glutamic acid replaced by valine. So this is a point mutation that brings about sickle cell anemia. They are protected. Why protected? Because that particular sickle-shaped cell is not allowing or accommodating the parasite inside. But they are not lucky enough, because anyway they are suffering from the severe anemia and they are having limited number of days in their life. But this type of persons who are producing G6PD, they are normal, they are having this particular malarial infection where glucose is converted to phosphogluconate. But there are certain people who are producing G6PD deficient or who are not producing G6PD. These people are also resistant. Why? Because normally the ROS or reactive oxygen species are not removed or sequestered. So because of this ROS, this particular malarial parasite can be actually checked. And there is another group. This is again something very interesting. There is a particular Duffy protein receptor, and this Duffy protein receptor is the gateway for the malarial parasite to come in. This receptor, if it is not developed or if it is modified, mutated in any way, then the malarial parasite cannot enter the RBC. And I told you, if it cannot enter the RBC, it will have to fast because its food is hemoglobin. So they cannot feed on hemoglobin, so they die, they perish. So even if the parasite is inside, but because of the absence of the Duffy protein, they cannot go in and they die. Okay? So these people are also resistant. There may be, I was telling that malaria can be detected in different ways. It is because of the antibodies. So what is the mechanism? You see, the moment these B lymphocytes are active, this B lymphocyte will have the merozoite protein or merozoite antigen. They will activate a particular HLA2, and this HLA2 will bind with this merozoite protein. So if we can measure the amount of HLA2, what is the amount of this particular antigen with a specific antibody, we can easily detect, or we can easily state that in what way we can detect malaria. So one way of detecting malaria is the antigen antibody reaction between the HLA2 and the CD4, and this HLA2 is combining with the merozoite protein. And ultimately, when this particular B cell will be having this merozoite, it will remain there for some time, but not for a long time. It will have to come out in blood and invade another RBC and feed on them. Ultimately, they have to go to the RBC. Only temporarily they can remain in the WBC, and when they are inside the WBC, you can detect them by this antigen antibody mechanism. And this mechanism, you can see clearly, this is a positive result because these antibodies are there, this is binding with the specific antigen, and you can say that this person is having malarial parasite in his blood. So this mechanism is called rapid detection test or rapid immunological test for detecting malaria. But of course, I told you, everywhere you may not have this facility of RDT. So what you do is you will check the slide, and checking the slide you can actually stain this malarial parasite, and you can differentiate between the structures of Plasmodium falciparum, Plasmodium ovale, or Plasmodium vivax, or Plasmodium malariae. And if he is a very experienced pathologist, he will immediately tell you what is the type of pathogen that has entered your blood. Now we come to a very important aspects of preventing this malarial disease. Ideally, we should prevent malarial disease by checking the pathogen through their vector. So you prevent mosquito So what should be the ideal way? Mosquito net. You can use mosquito repellent. You can use closed window panes so that mosquito cannot go in, because most of the time, let me tell you, they are in the evening, they are coming inside your house and they are feeding on the blood, because all through the night they will metabolize that blood, and you can see very often mosquito, their tummy is full with blood and they are actually resting in a place, and at that time they are utilizing that protein. So if you can prevent that, ultimately it will be reduced, the malarial risk will be reduced. And there can be pyrethrin, which is actually an insecticide that kills mosquito from pyrethrum flower. So this is a chemical control. And apart from that, there may be physical control, cultural control. What is cultural control? You will not allow the mosquito breeding places. You will not allow accumulation of water. What is physical control? You will make a barrier. It can be mosquito net, or it can be any sort of layer which will prevent the entry of mosquito or biting of the mosquito. And similarly, you can also have awareness. Many people doesn't know. If I ask you how malaria is caused, many people will say that it is caused by mosquito. But actually they don't know it is caused by the Plasmodium inside the mosquito. So this particular phenomenon has to be taught to other people. And this is also the duty of the government, and they may make people aware by observing the National Malaria Month, which is the month of June. Why month of June? Because more rain, more water, more malaria, right? Now you see, structure of quinine. Quinine comes from cinchona plant. Quinine is not the only drug. If I ask you how is malaria controlled, you will say controlling vector. But if the mosquito has already bitten and already there is infection, then you have to take drug. But without testing blood, if you take a drug, naturally it is not doing good to you, because it will destroy the liver cells. You will suffer from disorder of liver. So you have to test the blood. You have to be rest assured you have malaria, then only you take quinine. And these drugs are of this type, chloroquine, primaquine, and sometimes, this is very important, sometimes people already take it as a prophylactic measure. They don't have malaria, but they take this drug so that malaria doesn't take place. So these are one group of drug which are quinolones. There may be sesquiterpenes, there may be naphthoquinone. So artemisinin, quinine and atovaquone, these are the main drugs used against malaria. And there is a typical regimen, meaning, say for example, if you use the same drug for a continuous period, that drug will be of no use. So normally the regimen is like this. You start with chloroquine. If chloroquine is not there, you get into primaquine. If primaquine is not there, you get into metacalfine. Metacalfine is not there, you get into other drugs like artemisinin or artemisinin derivatives. And some of these drugs actually are toxic. So you cannot take a toxic molecule for a prolonged period, because that will be having a negative effect on your liver, on your general health, because liver is the metabolic king, I told you. So this is a very important drug, the benzyl menadione, which is preventing both. It is preventing the sporozoid development in the blood, and at the same time it is inhibiting the hemozoin formation, or it is inhibiting the metabolism of hemoglobin. So in both ways, the malarial parasite is in a fix and it perishes. Now we come to a very important aspect. What is that? The aspect of artemisinin resistant. Whenever we have a particular drug, a mosquito or it is, parasite will develop resistance. So that drug cannot percolate inside. So what to do then? Naturally, we have to prevent the disease. Whether we should go for vaccination or not to prevent the disease. Now what can be the possible way? The possible way is we take, this is the malarial parasite Plasmodium falciparum, from where we are taking the specific surface protein, we are inserting it into a virus. This is a virus body, and this virus body is inserted into the normal cell where they are producing this huge amount of malarial vaccine protein, and this virus is non-harmful to human. So this is the basis by which we produce a particular antibody against this known Plasmodium falciparum protein, right? And naturally, whenever there will be a new antigen, that particular antibody will prevent its replication. So that way the vaccine can be a very effective means. But that always this vaccine may not work. Now why it doesn't work? You must understand, basically this is binding of a sporozoid with a gateway protein. That is the point through which it will enter. And if the gateway protein is some way is blocked, then the virus cannot enter and it will perish. So this is the working principle of the malarial parasite. But the question is, this parasite is having multiple stages. It is having the sporozoid, it is having the hypnozoid, it is having the cryptozoid, it is having the merozoid. All these stages are different. And not only that, replication rate is also faster. So there is a problem of the titer. If the titer is not enough, malarial parasite is replicating faster, then it will overgrow. At the same time, sometimes new protein is there, you have answered to one protein, but if some other protein is there, it will enter. You cannot prevent it. So that is why the efficacy of the vaccine is questionable. But still, research is on, and there will be some vaccine which will be effective in the long run, of course in future. So I shall conclude the control strategy. At the outset, we should control mosquito. We will prevent their breeding. We take a pledge that we will not allow water accumulation anywhere, because that water is the breeding place of mosquito. We can have biological control by, say, by using some anti-mosquito protein in the form of Bacillus sphaericus toxin. We will kill that mosquito larvae. We can also have physical barrier, curtain or net by which we will prevent the mosquito entry. We can use chemicals by which mosquito biting is not possible. And at the same time, even if after all these things we are suffering, we get into the regimen of using chemical drugs, and these drugs we don't continue the same drug. We will vary the drug. Initially we take chloroquine and primaquine. Then if it is not working, we go to artemisinin. So in this way, we do not really allow that particular malarial parasite to be exposed to a compound, because you know, when a particular malarial parasite is entering our body, it is residing for quite some time within the liver, and that is the reason why if you are suffering from malaria, you will not be allowed to donate blood for the next 6 months, because even if the symptoms are not there, you never know, the malarial parasite is very nicely located in your liver. So students, I have discussed the different aspects of the Plasmodium, that is the malarial parasite. I have discussed the historical aspects of discovery. I have discussed what are their life cycle, in what way they actually cause damage to different organ. I have also discussed the life cycle stages and in what way they are propagating. And finally, I have discussed the different means by which we can control the disease. Thank you.